Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Desmosomes, also known as macula adherens, are the major cell-to-cell adhesion complex in the epidermis. Here I am telling major means some other minor adhesion complexes are also there. What are there? TAG, tag, tight junctions, adherents, and gap junctions. These are all minor attachments in epidermis. Coming to our major epidermal adhesion complex, desmosome. This desmosome anchors opposing keratinocyte cell surface membrane to the intracellular keratin intermediate filaments. Opposing means adjacent or neighboring keratinocyte. Opposing means mainly having opposite action. Here our keratinocytes all have same action. It connects opposing keratinocyte cell surface membranes to the intracellular keratin intermediate filaments. Means tonofilaments, also known as tonofilaments. These are main cytoskeletal structures of keratinocytes, nothing but keratin intermediate filaments. Hence bridging adjacent keratinocytes, thereby maintaining epidermal integrity, which is required for protection against physical, mechanical, and microbial insults. If the epidermal integrity is maintained, integrity means tight connections between the cells of the epidermis, it will protect us from external physical, mechanical, and microbial insults. Coming to location, desmosomes are located in all epithelial tissues. Desmosomes are located in all epithelial tissues. Isoforms of desmosomal proteins may vary in different epithelial tissues and in same epithelia also. Desmosomes create a 3D scaffolding of proteins. Scaffold protein means a protein which doesn't have any enzymatic activity, but it will bind to two or more other proteins and forms a structural support to the cell. Here desmosomes have three-dimensional scaffolding of proteins which fall into three major categories or three major families. First family, desmosomal cadherins, which contains desmoglein and desmocollin. Desmoglein Dsg, desmocollin Dsc. Next, second family, armadillo family. Example, placoglobin, placofilin. Last family, plakins. Desmoplakin, envoplakin, and periplakin. This envoplakin and periplakins are present in stratum corneum in corneodesmosomes. In entire epidermis, desmosomes are intercellular connections except in stratum corneum. Here, along with desmosome, corneodesmosin protein will incorporate and forms corneodesmosomes, which contains envoplakin and periplakin also. How to remember all these three families? Desmosomal cadherins starts with the word desmo. Desmoglein, desmocollin. I purposefully wrote here these two are pair, so I wrote them together. Next, second family, armadillo family, and last family. Both these families contain the word plakin. How to remember? Armadillo family starts with alphabet A, so put the word plakin first. Placo, globin, placofilin. Armadillo has the word O also at the end, so placoglobin and placofilin. Coming to last family, plakin. Hence it is last family. Keep this plakin word at last. Desmoplakin, envoplakin, and periplakin. This armadillo family and plakins families are called desmosomal plaque proteins. Why? These two are present intracellularly in desmosomal plaques. This desmosomal plaque is divided into two plaques, inner dense plaque and outer dense plaque. We will see it later. Now if these two families are desmosomal plaque proteins, then what is desmosomal cadherins? These are transmembrane glycoproteins, which means it crosses the membrane and will be present both extracellularly and intracellularly. Every protein will have amino terminus and carboxy terminus. Here, amino terminus of desmosomal cadherins binds to opposing desmosomal cadherins in intercellular space. This is extracellular portion of desmosomal cadherin. Now coming to intracellular portion, carboxy terminus, nothing but present in cytoplasm. It binds to placoglobin. Remember, first family will bind to second family will bind to third family, and third family ultimately goes and binds to keratin intermediate filament, forming a structural support. Here, first family amino terminus binds to opposing desmosomal cadherins, desmoglein, desmocholine of opposing keratinocyte. Then carboxy terminus comes and binds to second family, nothing but placoglobin. They also engage in both homophilic and heterophilic interactions, which means if same protein interacts with the same protein, desmoglein to desmoglein, desmocholine to desmocholine, this is called homophilic interactions. This is minor. Coming to major main interaction, heterophilic interaction, which is major interaction for intercellular connections. One protein interacts with other protein, desmoglein to desmocholine, exists as a pair. There are 4 desmoglein and 3 desmocholine isomers, each with varying expression patterns, both within and among epithelia. Within means within the same epithelia, among means among different epithelia, shows variations in expression. Coming to same epithelia, normal human epidermis. Here, remember like this, desmoglein 1 in first layers of skin, first isoform in first layers of skin, along with the desmocholine 1, first isoform of desmocholine in superficial layers of epidermis. Next, desmoglein 2, desmoglein 3, desmocholine 2, desmocholine 3 in deep layers of epidermis. Mainly desmoglein 1 and desmoglein 3. 1 is present and throughout the epidermis, but more densely it is expressed in superficial layers. Now only alone one, isolated one, desmoglein 4, it is present in superficial layers. If an extra PG is present, which unit he will be allotted? First unit. Same here, it is left out, it is present in first layers of skin, superficial layer. Now coming to different epithelia, isoforms, different isoforms in different epithelia, among the epithelia. Already I told you, desmoglein 1 mainly in the skin, entire skin, but more densely in first layers of skin, first isoform, first layers of skin. Desmoglein 3 in lower layers of the skin and mainly entire mucosal epithelia in oropharynx, expressed more, major, mainly, entirely in oropharynx, epithelia of oropharynx, and 1 and 3 is present in thymus also. In squamous cell carcinoma, desmoglein 3 expression is more, so it is being used as a target for molecular therapy to treat squamous cell carcinoma, desmoglein 3. Coming to desmocholine 1, along with desmoglein 1, it is present in skin. Desmoglein 2 and desmocholine 2, remember like this, 2 plus 2, 4 chambered heart. Defect, genetic defect in this desmoglein 2 or desmocholine 2 leads to autosomal dominant arrhythmogenic right ventricular cardiomyopathy. Remember, autosomal dominant. In some other place, autosomal recessive will come. Then coming to desmoglein 3, present in skin and oropharynx. Same like desmoglein 3. Desmoglein 4, present in testis, prostate, hair follicle. Genetic defect of this leads to autosomal recessive monolatrix and hypotrichosis. Autosomal recessive genetic defect. Desmoglein 4. Coming to Desmoglein Compensation Theory. This Desmoglein compensation hypothesis is proposed to explain the varied symptomatology of pemphigus vulgaris and pemphigus foliaceus. In mucosal epithelium, Desmoglein 3 is expressed entirely and dominantly, I already told you. And Desmoglein 1 is also expressed entirely but have very less expression. And skin, already we know Desmoglein 1 is expressed entire skin but more dominantly over superficial layers of skin. First isoform, first layers of skin. Desmoglein 3 in lower layers of epidermis. So now in pemphigus foliaceus, remember the word foliaceus for first isoform. Nothing but Desmoglein 1 will be damaged. What will happen if it damages? Desmoglein 3 is present entirely and predominantly in oral mucosa, so it saves the oral mucosa from blister formation. So there will be no oral lesions in pemphigus foliaceus. Here in skin, it is the dominant form. So there will be damage and there will be compensation by Desmoglein 3. Up to what extent? Only in lower layers of epidermis. So there is a possibility of forming blisters in the superficial layers of epidermis, leading to subcorneal split in pemphigus foliaceus because of damage to Desmoglein 1 target antigen. Here in staphylococcal scarlet skin syndrome and bullous impetigo, same Desmoglein 1 is the target of proteolytic cleavage by its exfoliative toxin, not antibody mediated but toxin mediated. So same, no oral lesions, only subcorneal split in epidermis. Coming to pemphigus vulgaris. What happens in pemphigus vulgaris? Mainly Desmoglein 3 will be damaged. Desmoglein 1 is already expressed in very low concentrations, so it can't compensate. It can't save the mucosal epithelium from forming blisters, so only oral lesions will be present. Why no skin lesions? Here Desmoglein 3 present only in lower layers of epidermis. So if it is damaged, compensation by Desmoglein 1 will be there because it is present throughout the epidermis, more dense in upper layers. So skin will be safe. No skin lesions. If only Desmoglein 3 is damaged, only. If Desmoglein 1 also gets damaged, then pemphigus vulgaris will lead to suprabasal split. Usually it will happen later. First oral lesions will be present, then skin lesions will also develop like suprabasal split. But as of now, in Desmoglein compensation theory, if one Desmoglein is gone, other Desmoglein will compensate and saves the skin. But it can't save mucosal epithelia and leads to oral lesions in vulgaris. Coming to applied aspects of desmosomal catharins. Nothing but Desmoglein and desmocollin. Already we know all isoforms. Now coming to first isoform, Desmoglein 1, weighing about 160 kilodaltons. I already told you, remember first isoform for first, F for foliaceus, pemphigus foliaceus. Along with that, pemphigus vulgaris. First majorly it attacks Desmoglein 3. Later it can damage Desmoglein 1 also. Paraneoplastic pemphigus. It has many target antigens, out of which desmoglein 1 is also targeted. Type 1 striate palmoplantar keratoderma. Type 1 isoform of desmoglein. Already I told staphylococcal, scarlet skin syndrome, bullous, impetigo, Netherton syndrome. In all these three, desmoglein 1 is the target of proteolytic cleavage, toxin mediated, not autoantibody mediated. Same. In type 1 striate PPK also, it is not autoantibody mediated, it is genetic defect of desmoglein 1. IgA pemphigus also attacks desmoglein 1. It has two types. One is intraepidermal neutrophilic type, another one is subcorneal pustular dermatosis type of IgA pemphigus. Intraepidermal neutrophilic target antigen is desmoglein 1. But in subcorneal pustular dermatosis, remember like this, subcorneal SC for desmocollin SC1, desmocollin 1. Coming to desmoglein 2 and desmocollin 2, I already told you, remember like this, 2 plus 2, 4-chambered heart, it leads to autosomal dominant arrhythmogenic right ventricular cardiomyopathy. Desmoglein 3, mainly in vulgaris, I already told you, pemphigus vulgaris. It is also affected by paraneoplastic pemphigus. Expressed in squamous cell carcinoma. It is being used as a molecular target for therapy to treat squamous cell carcinoma. Intraepidermal neutrophilic type of IgA pemphigus. Intraepidermal neutrophilic type, both 1 and 3 desmogleins. Subcorneal type, SC, Dsg1, desmocollin 1. Desmocollin 3, same like desmoglein 3, pemphigus vulgaris. Along with that, autosomal recessive hypotrichosis, and autosomal recessive monolatrix. Already I told you in desmoglein 4. First family finished. Then first family comes and attaches to second family, nothing but armadillo family. This family also localizes to nucleus, not only providing structural support, it also localizes to nucleus and modulates gene transcription and regulates cell proliferation. Coming to placoglobin, it is present throughout the epidermis, entire epidermis. Next, placophilins have 3 isoforms, same like desmocollin. Here 1 and 3 isoforms are present throughout the epidermis. Second isoform is present in the lower layers of the epidermis. Second family links third family, first family and third family. It links first family desmosomal catharins, carboxy terminals to third family, last family, desmoplakin amino terminus in outer dense plaque. Until here, outer dense plaque. Next, inner dense plaque. Only thing which is there in inner dense plaque is carboxy terminus of desmoplakin, which connects second family placoglobin to keratin intermediate filaments, structural cytoskeleton of keratinocyte. Envoplakin, periplakins are present in carniodesmosomes. I already told you. Here, how carniodesmosome is formed, it will be somewhat interesting. Our lamellar bodies will secrete protein carniodesmosin, which gets incorporated to desmosomes in stratum corneum. Hence, it got the name carniodesmosomes, which connects carniocytes. These carniodesmosomes are produced by lamellar body. Now, the same lamellar body will produce serine protease, means which cleaves the protein. To cleave and destroy this carniodesmosin, same lamellar body produces kallikrein 5 and kallikrein 7. In inactive form, it gets auto-activated, or with the help of matriptase, it gets activated, and its action is to cleave carniodesmosin. Then, now coming, again the same lamellar body will secrete lecty, Lekty, means lymphoepithelial Kajal type related inhibitor, which goes and binds to kallikrein and stops its action from destroying corneodesmosin. Now, if this lecti is defective because of genetic defect in SPINK5 gene, nothing but serine protease inhibitor Kajal type 5, there occurs Netherton syndrome. If no lecti, no inhibition of kallikrein, kallikrein will go and destroy all the corneodesmosomes. So, no connections between corneocytes. Corneocytes will peel off, leading to peeling of stratum corneum, seen in Netherton syndrome. Normally, these corneodesmosomes will be destroyed by kallikrein in superficial layers of the stratum corneum, leading to detachment of one by one corneocyte called desquamation. This desquamation won't be visible to our naked eye normally. If it is visible, it is called scaling. Nothing but visible desquamation, secondary lesion of skin. Where and all this corneodesmosome is present along with stratum corneum, inner root sheath of hair follicle. So, defect in corneodesmosome causes autosomal dominant hypotrichosis simplex of scalp. Autosomal recessive hypotrichosis in Desmoglein 3 and Desmoglein 4. Here, autosomal dominant. Here in punch biopsy of scalp lesion showed corneodesmosin aggregates around the hair follicle and loss of cohesion between corneocytes in inner root sheath of hair follicle. Coming to applied aspects of second family and third family. Second family contains placoglobin, which is present in entire epidermis, same like desmoplakin. Last family, it is also present in entire epidermis. Here, defect in placoglobin leads to Naxos disease, which have diffuse PPK, woolly hair, and autosomal recessive right ventricular cardiomyopathy. There, Desmoglein 2 and Desmocollin 2, 2 plus 2, 4, autosomal dominant. Here, recessive ARVC. Lethal acantholytic epidermolysis bullosa. Same in desmoplakin defect also. Lethal acantholytic epidermolysis bullosa. Epidermolysis will be there obviously. Along with that, anonychia, alopecia, no nail and no hair, neonatal teeth. Usually no teeth will be present in neonates. Here, neonatal teeth will be present. It is autosomal recessive. Next, placophilin 1. It is also present throughout the epidermis. Defect leads to ectodermal dysplasia, skin fragility syndrome, autosomal recessive. Which form of inheritance is dangerous, recessive or dominant? Recessive. It is very rare to occur, but if it occurs, it is very dangerous. Placophilin 2 present in deeper layers of epidermis leads to autosomal dominant arrhythmogenic right ventricular cardiomyopathy. Remember, wherever the numerical 2 comes, Desmoglein 2, Desmocollin 2, placophilin 2, autosomal dominant form of ARVC will occur. In desmoplakin, Carvajal syndrome, which is similar to Naxos syndrome except left ventricular cardiomyopathy. In Naxos, right. Paraneoplastic pemphigus. Envoplakin, periplakin in between corneocytes. Intercellular connections in corneocytes in stratum corneum. Paraneoplastic pemphigus. Here, so many times paraneoplastic pemphigus repeated. Where and all it's repeated? In first family, Desmoglein 1 and Desmoglein 3. In last family, entire last family. Last family means plakin word will come at last. Envoplakin, periplakin, desmoplakin. So, first family and last family, paraneoplastic pemphigus. Coming to structure of desmosome. With this table alone, we can draw the structure of desmosome and label each and every part easily. Remember like this line. Imagine when I am telling about this desmosome, it will be easy to label each and every part of the diagram. Remember this line as plasma membrane, this box as intercellular space or extracellular portion of desmosome, which contains only one part, amino terminus of desmosomal cadherins, both desmoglein and desmocollin. What it will do? It will connect opposing keratinocyte desmoglein desmocollin to second family plakoglobin plakophilin via carboxy terminus. These lines indicate crossing the plasma membrane because it is a transmembrane glycoprotein. Now here we entered intracellularly. Intracellular portion is divided into two dense plaque proteins, outer and inner. Here the word outer is there, but it is intracellular portion only, outer dense plaque, 10 to 20 nanometer away from plasma membrane. Here inner dense plaque, 40 to 50 nanometers away from plasma membrane, deeper into the cell. Here outer dense plaque contains already carboxy terminus came and attached to second family plakoglobin plakophilin. Entire second family is within outer dense plaque. Then it will bind to third family, desmoplakin amino terminus. So what and all things are there in outer dense plaque? Carboxy terminus of first family, entire second family, amino terminus of third family, nothing but desmoplakin. Now this amino terminus of desmoplakin connects second family to keratin intermediate filaments via carboxy terminus. The only thing present in inner dense plaque is carboxy terminus of desmoplakin, which attaches to keratin intermediate filaments. Now we will see the diagram and we will label easily. Remember this as intercellular space. Fluid accumulation in intercellular space is nothing but spongiosis. First desmosomal structure is identified as only connections left in a spongiotic tissue in punch biopsy. Now here our first family, desmoglein and desmocollin heterophilic interaction, transmembrane glycoprotein, it crossed the plasma membrane and linked with opposing keratinocyte desmoglein desmocollin. Now it goes and links it to second family plakoglobin and plakophilin, which in turn goes and links to amino terminus of last family desmoplakin. This entire thing is outer dense plaque intracellularly. Now the last family goes and connects to keratin intermediate filaments. Carboxy terminus of desmoplakin is the only thing present in inner dense plaque of desmosome. The entire structure of desmosome finished. Now we will see destruction of desmosome, nothing but acantholysis. Why this word came as acantholysis? Because in stratum spinosome layer, the desmosomes are known as spine-like structures, nothing but acantha. Lysis of this acantha means lysis of desmosomes only. This is coined by Auspich. There is another sign named from him, Auspich sign in psoriasis. Coming to definition, nothing but loss of coherence means connections between epidermal cells due to breakdown of intercellular bridges. Already we saw desmosomes bridging opposing keratinocytes. Pathogenesis. Initially there will be separation of interdesmosomal region, means one desmosome will get separated from another desmosome. Then there will be splitting and disappearance of desmosomes. Intradesmosomal proteins will get splitting and disappearance, forming complete separation of one keratinocyte from another keratinocyte, leading to intercellular space. This intercellular gaps will lead to fluid influx from dermis, leading to cavity formation. Cavity may form intraepidermally only, but according to level of split, subcorneal or granular layer, spinous layer or mid-epidermal, suprabasal split, according to level where it divides the cell. in the epidermis. Coming to triggers of acantholysis, already we saw autoimmunity means autoantibodies targeting desmosomal proteins, genetic defect in desmosomal proteins, infections, already we saw staphylococcus, scarlet skin syndrome, bullous impetigo targeting desmoglein 1 and cleaving it, infestations. What is the difference between an infection and an infestation? Infestation means if an parasite enters our body, it is called infestation. If microorganisms like virus, bacteria, fungus enters our body or infects us superficially over the skin also, it is called infection. Ionizing radiation, UV radiation, specifically UVB, burns, heat, friction, all this leads to sweating, ultimately maceration, trauma to the skin. Whatever damages the skin may lead to secondary acantholysis. Dermatitis, drugs and foods containing thiol group like penicillamine, garlic and onion. Phenol group, phenol, phenobarbital, aspirin, rifampicin, cephalosporin. Phenols and tannins containing foods like nuts, banana, tomato, mango. Non-thiol group containing drugs as inhibitors and glibenclamide. All these are trigger factors for acantholysis. Acantholysis is of two types, primary and secondary. Primary means dissociation, disintegration of desmosomes either due to direct injury or due to hereditary defects in construction of desmosomes. Here the primary event and the only event is acantholysis, which leads to separation of keratinocytes, forming gaps, fluid influx from dermis, and ultimately intraepidermal cavity formation. Primary acantholysis. Examples: pemphigus group, Hailey, Darier's, Staphylococcus, scarlet skin syndrome, bullous impetigo, transient acantholytic dermatosis. Coming to secondary acantholysis, in name itself it's there, acantholysis secondary to damage to keratinocytes by various factors. Here the primary event is keratinocytes injury, later leading to subsequent dissociation and disintegration of desmosomes. As a consequence, secondary acantholysis. Example: herpes zoster, herpes simplex virus infection, genitalis and labialis, varicella zoster virus infection, varicella, epidermolytic hyperkeratosis. If you remember until here, it's enough about acantholysis. If you want some more examples of secondary acantholysis, acantholytic Dowling Meara type of epidermolysis bullosa, lethal acantholytic type of epidermolysis bullosa. In lethal acantholytic type, there will be defect in desmoplakin and placoglobin. Already we saw both of them are present through entire epidermis. Next, acantholytic acanthoma, acantholytic Dowling-Deago, also known as gully disease, adenoid squamous cell carcinoma, basal cell carcinoma, BT leprosy, condyloma acuminata. A, B and C. Malde-Malida type of PPK, Pityriasis rubra-pilaris, psoriasis, tinea corporis, whatever the disease causes chronic inflammation and leading to skin damage, ultimately may lead to secondary acantholysis rarely. These are all rare examples, optional to remember or not. Remember my channel name, The Average Dermadoc. Don't forget to subscribe. Thank you. Hasta la vista. Dasvidania.